Thank you all for joining. I'm Martina. I'm a part of Project REDDIE, and we are basically focusing on real-world evidence for decisions in diabetes. Today, we will have first of our many webinars where we will focus on the work that we are doing in Work Package 4, and you will learn more about that soon. Today we have two speakers, our work package leader, Professor Christian Torp Peterson, and also we have Thomas Alexander Gertz from the same group that will explain more about the methods that we are using. Just before we start, here are some housekeeping rules, some technical things. So if you have any questions, we will have a designated time section at the end for the Q&A, and you can leave all the questions or comments that you have in the section in Teams for Q&A. If you have any technical issues, please feel free to ping me in the chat, and I will try to assist you. Then you will have the recording, and this will be available also on our website, and you will also receive a link. So feel free to just enjoy and listen to us. And of course, please be respectful for our speakers and fellow participants at all times. We really encourage open and constructive dialogue within this webinar. So just a bit of an information about REDDIE. We have 15 partners from really different institutions, universities, patient boards, regulatory bodies, and so on that help us really to navigate this area of RVE and RVD. And our key objectives we kind of put in a question, which I will read now. So to help people with diabetes, how can regulators, payers, and guideline developers augment data from randomized controlled trials with data from other than RCTs to make decisions? And we are doing this to, of course, support the use of RVD to make it more efficient, more cost-effective, and of course, safer. Today, you will also hear how we are using this method that we are working on these 4 large national registries as well. And of course, we will have more webinars on the results as well. So keep tuned in our social media as well. Then we are also using machine learning. We will also have a webinar at that part. And in the end, we will share more about the standards that we combine from all of this analysis and thinking, and we'll see how this will help the final evaluation. Today we're focused on the analysis that we're working on within Work Package 4, and we are using 4 registries from England, Denmark, Sweden, and Germany, and also some other data, but Christian will definitely tell you more about that. And for that purpose, I will stop sharing my screen and tell you more about Christian. So he is, of course, our leader of a work package, but also he is a professor of public health and cardiology at NOH and the University of Copenhagen. He has conducted research in randomized trials as well as outcome studies using nationwide registry data. The publication list that he has spans from a wide range of subjects, including heart disease, hypertension, heart failure, cardiac arrest, and diabetes. And he has authored over than 1,200 international publications and holds an H index of 154. But let me give the word to Christian, and I will share his slides now. There you go. Christian, whenever you're ready.
Well, thank you. Thank you very much for the kind introduction, Martina. This is a work by Martina Kokoni, Greek artist working in Glasgow. I'm showing it because it is a modern still life. And the reason I sort of show a still life for a beginning comes with the next slide, please. This is the summary message from an extremely important paper that Miguel Hanan and Robin wrote in 2016, where they introduced the concept of trial emulation and how it just differs from good epidemiology. And the major features they suggested was that you could emulate trial in real-world data if you followed a strict list of points. You had to have clear eligibility criteria for entering a hypothetical study. The treatment strategies should be clearly defined, assignment procedures defined, the follow-up period should be defined, a clear outcome, and then you should define a causal contrast of interest in an intention to treat effect or a per protocol effect. And then you should publish an analysis plan. The reason I showed a still life was that they used start of therapy as a sort of analog to intention to treat in a clinical trial. And that is a still life. It ignores what happens in the future. Next slide, please. This is some reanalysis of the LEADER study, a pivotal study to show GLP-1 efficacy in high-risk patients with diabetes. And this shows what intention to treat was in this trial on the left slide, where you see that really a very high proportion of patients were loyal to liraglutide or to placebo. On the right, we have real-world data from a Danish register of medication, and we show diabetes patients that start on either GLP-1 or in this case, and I'll come back to that, a DPP-4 inhibitor. And what you see is that within about a year, 20% have had a break in therapy. And what this clearly shows is that starting treatment in real life is not the same as intention to treat in a trial. Next slide, please. Now, what can we do about this? Well, one of the newer techniques, and it's very new to medicine and to mathematicians, is just some of it is just 10 years old, is that you can do a technique called longitudinal target maximum likelihood estimation. This is a technique which is primarily developed at Berkeley University in the United States, led by Mark van der Laan. Next slide, please. Now, with this slide, I'm trying to put this method on one slide, and that is actually summarizing the content of 2 full books on one slide. So it has to be very superficial. Now, the first thing about longitudinal target and maximum likelihood estimation, or LTMLE as I'll call it, is that you need to define a treatment strategy. And that is, for example, X years of continuous therapy with A versus continuous therapy with B. Once you've defined that and people can start on those therapies in your population, you need to select the population appropriately and then build a complex prediction model. And that needs to adjust both for baseline and time-dependent confounders, how they influence outcome. That is sort of the technique that is done in classical regression, but in this case, we also take into account changes over time. And then further, you should adjust for what baseline and time-dependent confounders do to exposure. Doing these both is called double robust. Finally, included with this is a use of flexible machine learning to adjust for confounders. Once you've built this very complex prediction model, we'll use it to make a G-estimation, which is a highly counterfactual, hypothetical trial where we expose the whole population to strategy with treatment A, and then afterwards again with strategy with treatment B. This analysis of continuous therapy can be looked as an idealized per protocol as opposed to intention to treat. Next slide, please. Now, with this technique, we got a grant to do the REDDIE study, and in work package 4, it was agreed that 4 countries would establish this analytical platform using longitudinal LTMLE, and there's a lot of mathematical work going on to make it easier to use the method. Then we should compare how trials from the real world, real trials look when we emulate them in national registers, and further, how those who could not be in trials would behave. And then we should integrate the whole thing. And very important, this is done independently in Swedish, German, United Kingdom, and Danish registers. Next slide, please. Now, this is just to tell everyone that there is a highly increasing interest in looking at estimands for trials. While intention to treat in trials has been rather uniformly defined, then at least per protocol can have to a large extent been defined very differently in different trials, and it needs to become much more rigid in the future. Next slide, please. Now, the estimands we are using in the REDDIE study for GLP-1, we're doing several drugs, but GLP-1 is what we're starting with, is start liraglutide and stay on it for 3 years versus do not take liraglutide. That's the leader study. Now, if we were to emulate that in real-world data, we would need to emulate the discontinuation probability of leader in observational data. This we so far considered not feasible to do in a meaningful way. We cannot use starting therapy as a replacement. I've shown you that since the discontinuation in clinical practice is high. We cannot emulate placebo very good, because to get into a trial, you need to contact a doctor. And if we just match with people not on therapy, they don't necessarily need to be in contact with a doctor. So what we do in a trial is we emulate GLP-1 versus an alternative neutral therapy. The neutral therapy we've chosen for nearly all the studies in the REDDIE consortium is a DPP-4 inhibitor, where randomized studies have shown it to be neutral with respect to cardiovascular outcomes. So what we're going to do with the REDDIE study is to emulate GLP-1 treatment for 3 years while never taking DPP-4 versus use DPP-4 for 3 years and never GLP-1. Now, how do we compare this with the leader study? Well, what we do is we reanalyze the leader using LTMLE for comparison. Next slide, please. Now, this is the comparison with leader. To the right, you have almost identical to the published in New England Journal of Medicine. There are a few patients that were excluded from the data that were made available to us, but you see a result identical to what was published is that risk difference after 3.5 years of 1.5% in favor of GLP-1. To the left, we have reanalyzed the leader using LTMLE, and we get 1.94%. Now, and that is exactly what we would expect, a slightly higher effect, since to the left, we are emulating continuous therapy, while to the right, we are recognizing that some people do not stay on therapy. Then the next result, next slide, please. We're trying to see, can we emulate leader in a Danish national register? And what we have to the left is the main outcome of the LEADER study, a major cardiovascular event. And what we see after 3.5 years is a 2.5, which is slightly higher, but quite similar to what we found in LEADER. We also have a similar result with cardiovascular mortality. So we can say at this stage that we can, in Danish registers, successfully emulate the LEADER study. Now, the deadlines for producing all these comparisons is quite soon, and results are pouring out at this time. Several others are looking very good. But next slide, please. We are not only considering dichotomous outcomes in the REDDIE consortium, also hemoglobin A1C outcome is studied, and this outcome is estimated in a population where we actually have censoring and we have competing risk. And this causes some challenges because of right truncation of data. And my last slide, Thomas Geertz will in the following presentation in particular address some of these issues. Thank you for your attention. Are there any questions?
If not, we can also wait for the end and you can type your questions to Christian. But for now, Thomas, if you're ready, I will do a bit of an introduction to you as well. So the central topic of his methodological research is the development of statistical methods for clinical risk prediction models. He works also with advanced techniques for big data analysis, such as machine learning and cross-validation, as well as semi-parametric models for causal inference in longitudinal time dynamic settings. A focus of his applied collaborative research is the interpretability of estimands, which specific medical and epidemiological applications. He's particularly interested in the challenges posed by censored and missing data in the analysis of non-randomized settings, and additionally, part of his research time is dedicated to the development of user-friendly statistical software. So feel free to also maybe ask him about all of this interest as well. But now, Thomas, feel free to start your presentation.
Good. Can you see my slides?
Yes.
Good. Okay, thank you for the introduction. I will now discuss very much the same as Christian, but from a more statistical detailed perspective. To begin with, just to remind you of the definition of an estimand. So the estimand is the target of inference that we define before collecting the data. And the estimand should have the attribute that if we would know the value of the estimand, then we could answer our research question, our clinical question. So that is important to distinguish between the estimand and the estimator, which is the method that we're actually using in order to get a numerical result, which we then call the estimate. So this is all very confusing, but we have to be very strict here in order to not get confused. So an example that we're working with in the REDDIE Work Package 4 working group is the difference in the average 5-year or 3-year probabilities of a cardiovascular outcome if everyone would take GLP-1 for 5 years versus if everyone would not use GLP-1 for 5 years. And here we have to distinguish between the randomized trial world, where we have a placebo group which does not take GLP-1, whereas in the observational data we do not have a placebo group, but we have to work out with an active control. So here we have been using the DPP-4 as a control group. But it's also true that in the randomized trials that are conducted in these times, the placebo group will still use the best care apart from the active group. So they are not allowed, and the people in the control arm are not allowed to take the GLP-1, but they are allowed to take, for example, SGLT2. And then, as you may know, in calendar time, SGLT2 was released on the market later than GLP-1. So even in the randomized trials that they do on GLP-1, they have to accept that in the placebo group, people will start taking SGLT2, which is a very good competitive drug as well. So all these are challenges which, as I try to explain, are not only in the observational world, but also in the randomized trial world. We have the same type of challenges. There's, of course, still more control in the randomized trial world, but we have to be aware that there are the same type of problems also on that side. And then another motivational slide here, what if, that's the causal inference type of question. And here I'm asking, what happens if we do not define the estimand before we do the analysis or even before the estimator? So there's a large volume of literature where people develop methods where they get a result at the end, but they have a hard time explaining what the interpretation is of the result. So this is this old style of statistical analysis. You have taken to account what is the outcome. The outcome is survival. Okay, I do Cox regression because that fits to survival, and then I get a hazard ratio, and then I don't know maybe what it means. And here's an example from paper by Swisser, where they have done some analysis where they do some time conditional propensity score matching. They also do some other prescription-based exposure sets matching. And in both cases, they get a hazard ratio of heart failure with GLP-1 use versus SU use. And the problem is, what does this value here, 0.373, mean? So I say that this doesn't have a clinical interpretation for a person, and it's also very difficult to understand what is the population in which this parameter has an interpretation, and moreover, what is the follow-up period? How long did people take GLP-1? All this is not defined, so this is not very interpretable result, and that is the danger of not defining the estimand before defining the estimator. When we now focus first on the randomized clinical trial situation, here we need to start by saying at what time we will enroll patients or people, and then we define usually some inclusion criteria. And once this has been done, then one needs to write down the treatment protocols which dictate the treatment throughout the follow-up period. So for example, we could say use GLP-1 for 5 years, but do not use DPP-4 for 5 years. That would be a definition of a protocol which dictates what the people should use when they are randomized to this protocol. It is most of the case important that you also already, before doing the starting the trial, think about possible contraindications and side effects. So if a person who is randomized to GLP-1 has severe side effects such that the doctor says you need to take a treatment break or you need to stop taking this treatment, then quite likely this can be foreseen in the planning phase and should be introduced into the protocol in order to get a sound definition of an estimand. The problem is that rescue medicine, for example, a single shot of insulin, whether that is allowed or not in the treatment protocol would alter the interpretation of the estimand. Okay, then once you have defined the protocols of your different arms or regimens that you have in your trial, you randomize people to one of them. And then we need to define the estimand. That could be the expected outcome if everyone complies with randomized protocol. But in a follow-up study, this would also require some time horizon. So we need to say the estimand is, let's say, the expected outcome after 3.5 years or after 5 years after the start of the study. And then one needs to specify in all details how to estimate the estimand. And here, one possibility also in the randomized clinical trial world would be to apply a longitudinal targeted minimum loss-based estimator. People would then say, why would I use that? I usually do the intention to treat analysis, but we have to be aware here of the problem that the estimand of the intention to treat analysis in the randomized clinical trial world depends on what the people in the trial do in terms of compliance to the treatment protocols. So people will deviate from the treatment protocol for different reasons. Some of these reasons should have been allowed for by the protocol. For example, rescue medicine or severe side effects would probably mean that the patient should have a rescue medicine or a treatment break. And if that's specified in the protocol, then there is no treatment deviation if it happens. But others must be forbidden by the protocol. For example, to forget the medicine. If the patient forgets to take the medicine, this must be forbidden. And if there are rather many people who forget to take their medicine, then of course the interpretation of the intention to treat result will be affected by this. And in any case, it's nice to have a pre-specified estimand, something that does not depend on what happens during the trial. And as such, this requires an estimator such as the LTMLE, and it also requires collection of time-dependent variables about the indication and reasons for violation of the treatment protocols. And that is also the case for the analysis of randomized clinical trials. Now, switching over to the emulated target trial world that we are doing in the registered data in various European countries in this work package, the first step here is to specify the ideal randomized trial that we would like to do but have not done, and define inclusion criteria and protocols and estimand as we would have done it if we were able to plan and do this randomized trial. Then we need to look into our data, available data, and see how we can define and approximate all these things like time zero, the inclusion criteria, the treatment information about whether the patient takes the treatment and for how long, and time-dependent covariates and outcomes. We need to write down all the deviations between the emulated trial and the ideal randomized trial, because these will likely affect the interpretation of the results. And we would also need to write down all the limitations that we may have in our data and the assumptions that we make in order for our estimator to be to deliver an estimate of the estimand that we have defined. We use baseline and time-dependent variables to estimate the estimand, and here we use longitudinal target minimum loss-based estimation. We should mention here that people enrolled in the emulated trial, they're not really enrolled, they do not know that they participate in our emulated trial, and hence they are non-adherent to the treatment that we invent. So with this protocol is defined by us, and these people don't know that they are participating, so they are deviating for the same reasons that people deviate in the randomized trial, but also for many other reasons, simply because we do not have information like the doctor says, okay, you should take GLP-1, and if you don't like it, then maybe after 6 months you should switch to DPP-4. This type of information we do not have available, and hence we have to be more careful when we analyze the observational data, because it's quite likely that there are many other reasons for people to deviate from the start treatment than what is the case in the randomized controlled trial world. The challenge for the estimator is quite nicely summarized here on this page. In the perfect world, everyone would be following whatever treatment protocol the person was randomized to, for example, using GLP-1 for 5 years. And in this perfect world, this person would also be fully compliant and attend all clinical assessments and would be followed for the exact amount of 5 years. Of course, if outcome happens before, then just until outcome. In the randomized trials in real life, people switch, stop, and restart treatment. They drop out, they immigrate. And these are all the tasks that the estimator needs to deal with. In addition, there may be missing doctor visits and other reasons for missing values in the variables that we are working with, and that's another problem. And then finally, there is truncation by death, and I want to spend some more time later on about the truncation by death issue. But first, let's recap here how this estimator works. So the longitudinal targeted minimum loss-based estimator is in fact a time sequence of regression models where both the models for the outcome and models for the propensity score and for the censoring are incorporated into one estimator. And it's a time sequence which requires a discrete time scale. So usually when we, for example, analyze 5 years, then we do half-year intervals. We could also do 3 months intervals or 1 month intervals. So this would still be feasible. But we would be required to create and estimate a regression model for the outcome, a regression model for the propensity of treatment continuation, and a regression model for the likelihood of dropout and administrative censoring at each of the time intervals. The estimator should estimate the estimand in this hypothetical world where there are no censorings, there are no missing values, and there are no violation of the treatment protocol. So the estimand is defined in the perfect world, and the task of the estimator is to apply weighting and regression in order to estimate the estimand based on the observed data. We need to note that truncation by death is different. So when a person dies, then we are 100% sure that the outcome doesn't occur after death. So that is very important to recognize. And anything else also does not occur after death. So we do not have an A1C value after the death, and so forth. So treating death as censoring or as a missing outcome, if the outcome is the A1C on a continuous scale, that would essentially lead to a study of life after death. And that is very, very, very rarely clinically meaningful. So we don't like it. Now, let's turn to how the estimator works. So the estimator specifies a regression model for the outcome. We start with the last time point, and then we ask the regression model about a prediction for a hypothetical person. Let's think about a clone of the current person, which is alike, but except for the treatment, because we set the treatment to the value of one of the protocols of our trial. So this is the general G formula estimator that Robbins has invented in the 80s. We have to be aware that when we do this, we are at risk of extrapolating in regions of the cohort where there are either no people or where there may not be people. Either there are no people observed in that area, or there are no people. And that extrapolation is of course more prone in parametric models than it is in machine learning models. But in both cases, we need to be aware of it, and we need to be very worried about that we do that. So extrapolation is not a good thing if there is a situation like the following. For example, a person's A1C value has been measured and it's very high, so the doctors decide to treat with insulin. Then if our protocol says it's not allowed to treat with insulin, then in these emergency situations, it may simply be that no one with such a high A1C value was not treated with insulin, and hence there's no positivity, and hence we cannot expect a person to be in that area of the covariance space. For the weighting techniques, the limitation is very much the same, but the idea of the weighting is that when we have a person who deviates from one of our protocols, then we use a propensity score model to give more weight to the people who are like this person, but still continue to take our protocol treatment. And then the idea is that by giving more weight to these people, they will also represent the person who deviated from the protocol. The technique for censored data is the exact same when immigration or end of register censors the follow-up of a person. Then the censoring models would put more weight on those people who are still uncensored, but otherwise alike the person. So here in the register data, here we have to be aware of the problem with the calendar time period. If there are trends in, let's say, the outcome or other things in calendar time, then then there may be violations of this principle, which requires that we are able to find people alike the person who are not yet censored. And more generally, there are positivity violations if we cannot find any or enough people who are like the person who was censored or deviated. And these type of problems are usually violations of the positivity assumptions, are hard to solve, and may even require a change of the estimand in order to accomplish a meaningful analysis. Now let's, for the remaining of my presentation, focus on the continuous outcome situation. I try to illustrate here what could be the measurements of A1C over time. Time 0 is the start of the treatment, and then time and month on the x-axis, and a blue curve is personalized for one person A1C trajectory, and we can see it goes down and then up again and down again and so forth. So it varies, and it has also irregular measurement time points, and they will be more or less irregular, more regular in randomized trials and less regular in registered data. The problem is that if we have missing values in such a trajectory here, illustrated by the question marks, then we don't have the measurement available. So maybe my graph is not perfect. So the first question mark seems to be correct, but for the second question mark, there should of course be a dotted line to the question mark because we do not observe that value. And then you can also see my trajectory here is truncated by death. And then this is of course both challenges. We can try to estimate the value at the question mark based on the values that we have for the other people, but we cannot, it makes no sense to estimate the values after the death based on other people, because that would be, the aim would then be to analyze what would happen after death. Now, when we have truncation by death, then we have to be, we can acknowledge that the longitudinal targeted minimum loss-based estimation can handle truncation by death if the outcome is a time to event variable, such as time to heart failure or time to MACE or time to all-cause death, time to cardiovascular death. However, truncation by death spoils the interpretation of the average A1C value, for example, after 5 years. There's a paper by Chetkin who defines the survivor average causal effect as the expected difference in average A1C, say 5 years after treatment start, in people who would survive for at least 5 years under all protocols of our emulated trial. And the problem is, this is a sound causal interpretable effect, but how interesting is it for the clinician? That's not so clear, because when you are with a patient at time 0, you do not know yet whether the person would survive under both treatment regimens. And in particular, if there's differential mortality according to the two protocols, then the interpretation will require that we also have an assessment of the differences in the mortality. An alternative to the survivor average causal effect would be to study simply the first time where the A1C trajectory crosses a predefined threshold, and that could be the diabetes threshold 48, or maybe even a personalized threshold, which would be like 20% of the person's value measured at time 0. So if you can reduce your A1C by 20% according to your baseline measurement, then you maybe have achieved the treatment goal, and you could say this is a positive result for the treatment. And if we define the time to the first crossing of the A1C trajectory as a time to event variable, then we can deal with a competing risk of death in the usual way that we have learned for the longitudinal targeted minimum loss-based estimator. And to illustrate this, I have the curve from before, the first one without missing values, because missing values would clearly give more trouble here. And we can see here the value to begin with is 68 and then 20% of 68, that's this dashed line here, horizontal line, the blue one. And then we can see the trajectory here if we interpolate between the measurement values, which may or may not be justified depending on how far away the two measurements, so this one and this one are. Here's where it crosses, so this would be the time, and months would be like something like 38. This would be the event time where the person has a treatment success in terms of having reduced the starting A1C value by at least 20%. We can then also see that later on the curve goes up again, but then this is clearly possible, and that's nothing we can avoid in this with this type of target parameter. Okay, to sum up what I've been presenting for the work package 4, we have been very careful and separated the definition of the estimands from the way the estimators are constructed based on the data. We are currently applying the longitudinal targeted minimum loss-based estimator, and in this way deal with censored data and deviations from the protocols in both the reanalysis of the randomized trials and also in the emulated trials. Missing values in the covariates can maybe be imputed by a super learner of the nuisance parameter models, and there I mean the outcome regressions, propensity score models, and censoring regressions. However, we do not have sufficient experience with this type of analysis, and I think we will first have to do more research on how the method performs when we impute missing values here of the covariates in the one or the other way. And final comment here is that truncation by death is mostly an interpretational problem. The expected outcome, whatever outcome it is, under given treatment protocols cannot be interpreted without also knowing the probability of death. Thank you for your attention.
Great, thank you very much, Thomas. Until our participants get a bit braver with questions, I also have some prepared for you and Christian. So Thomas, since you're the last to present, would you say, are there any specific types of studies or clinical questions where you would maybe not recommend using this method?
Wow, I'm not sure I understand that question.
Okay,
so
fair.
I can, if you tell me about a study that you want to do, then I can often think about it and come up with a suggestion on how to analyze the data. But if you ask me what is a particular method not useful for, then I think the answer is infinite.
Okay, great. And how do you see this method evolving within healthcare research? Do you see any maybe specific, let's say, benefits where you could maybe recommend using it more?
Well, we are now in the process of using it here across different countries, research groups. So this is a nice way of spreading the word. I have a feeling, I'm not sure how far they are, but the research group in Berkeley, they are discussing with the FDA about maybe using some of these techniques more officially, also forcing people to use them for the randomized trial. But clearly, I would like to see this method applied when, instead of what they call the per protocol analysis, where people simply delete the data from people who have not been following the predefined protocol. So simply deleting data is not a good approach and is prone to bias. So here I think this method is would have a nice place.
Okay, great. Thank you, Thomas. And Christian, a question for you. So within all the experience for now that we have in Project REDDIE, what would you say are the biggest challenges that we are facing in the analysis?
Well, the biggest challenge is making regulatory authorities trust what we do, because they have a tradition of being, to my taste, too rigid, only listen to something that comes from randomized studies. And the point is, we will never get randomized studies of everything. So there will be many situations where it's either impossible, unethical, or too costly to do a study, and there we need to use observational studies. And what I hope we help achieve with the REDDIE consortium is to show that we in 4 different national registers can emulate some studies correctly. Now, that is, of course, a secondary objective, sort of just in showing that the studies work, but where the real benefit is, how about all the patients that never entered into a trial? Are these treatments as effective in those, or are they not? That is, to me, the major result of the REDDIE consortium in clinical interpretation, and is also the situation where I hope and possibly even believe that regulatory authorities will use the methods in the future.
Okay, thank you. Since we don't have questions, which is also fine, I think also Thomas and Christian are open to any questions that maybe they receive via email or something. You can also reach out to us via REDDIE social media, and we can give them the questions as well. I would like to ask them if there are any final thoughts that they maybe have. If not, if so.
Well, one final thought is that you asked about where we're going to use this. We are expanding it to use in other situations. We are working with the Danish Cancer Register, and they are initiating some studies of development of cancer following several therapies. We are working with gynecologists to look at female hormone therapy, especially postmenopausal hormone therapy, what that does to cancer and cardiovascular risk, and we will implement the LTMLE method to do this. And so I do expect that the knowledge and use of this group of methods will increase a lot in the future.
Mm-hmm. Great. We also received a question in the chat from Francois. So not only regulators or HTA experts need to understand and trust the emulation method, patients would also need to understand and trust their results. Is REDDIE planning to explain these techniques to patients and their organizations? Actually, yes. So we have a patient advisory committee from really different people. So from young students to also people working on universities and in different other patient advisory committees. We are now working on all the results, and we also had a couple of meetings with them to kind of explain what we are doing, and they're really helping us to kind of shape the communication and also the results to something, you know, easily feasible and tangible to, you know, a regular person, because in the end we are using patient data. And we do see all the ethical aspects as well. So we will definitely do more communication-wise when we have all the results done and worked through with our patient advisory committee. You can also find them on our website as well. If you want to talk to some of them. We're also happy to, when we publish some of the things, and if you wish to maybe be a part of it, let us know, and we could work together on that. We do see the need, yeah.
I could add to that the reason such a question comes up is because the technique is new and is difficult to understand for doctors and patients. But for example, if you take the LEADER study, the main result of that is a hazard ratio. Now, how would a patient understand that? Take this drug because your hazard ratio is 0.8 or whatever it was. It is extremely difficult to understand. And if the patient asks the simple question, then doctor, how much longer will I live? Then the doctor is not able to answer that question. Actually, the LTMLE is more simple to understand because we say, if you go for this therapy and you maintain it for 3.5 years, you will have a benefit of about 2% in a probability of death or whatever we are looking at. That is it is possible to understand that. And further, using this absolute risk change is very important because it is not as exaggerated as hazard ratio usually is. And that's very important when the patient has side effects, because when side effects occur, it's very important that you have not overestimated the benefit of a drug, so the patient will tolerate unreasonable side effects in order to maintain the therapy. So I think eventually the LTMLE is easier to explain to patients than hazard ratio.
Great. Thank you, Christian, very much. Thomas, do you maybe have some final words?
No, I think I'm good.
Okay, thank you. Then thank you all for joining us today. We will have more webinars. One is coming up also this month. So feel free to still register if you want to. I left a link in the chat as well, and you can find it on the website as well. And thank you all for joining. You can also follow us on our LinkedIn and X account. And if you have any questions, feel free to reach out to us. We will also publish the recording, as I said, on the website. And yes, thank you all and have a nice day.